Okay, so here is the treatment portion for future directions. I'm going to go ahead and be discussing two approaches that I found most interesting. So the first approach is from the London School of Hygiene and Tropical Medicine. So as I mentioned before, researchers are really concerned with the fact that these worms can become resistant. So this is an important part. So praziquantel at a high dose is only effective on adult worms, they found. So it's not as effective in juvenile worms. So they say that this is why repetitive treatment is required, because this drug is only attacking adult worms and still leaving the juvenile worms alive. So they're looking for a drug that'll kill all worms in a smaller one-time dose. They definitely want to reduce the times that patients have to be on treatment, and definitely a smaller dose that they only need to take one time, and in comparison to the other drug that has to be taken 3 times and you have to do it 2 times a year. They definitely want to find something that's easier. What they did is they were screening over 300,000, and that's a typo, my bad, my mistake. They're screening over 300,000 compounds to discover any compound that has the ability to kill all worms, and they're looking just to find compounds that can kill both adult and juvenile. They said that this one is from last year, so right now they're still testing and doing clinical. Well, they said that they're still testing and that clinical trials are still needed, but I thought this was really interesting. Another interesting treatment case that I found is from the University of Texas Southern Western Medical Center. This one is more recent. This one was done in April of 2022, so last month. So because of the discovery that females need physical contact with males to become and stay sexually mature, they came up with a really interesting theory. So these females need to be with the male in order to stay sexually mature and then, lay the eggs. And these eggs are the ones that are responsible in causing schistosomiasis. So they looked at 2 genes that can affect the relationship that they have between each other, or just keep them from not being able to reproduce. So they looked into they started by looking into any changes in gene activity between the male and the female schistosomes when they come into contact. And the first one that they found was the GLI1 gene. And this one is pivotal in males to prompt female sexual maturity. So they also found that when this gene was removed, females could not reach maturity and then they couldn't lay eggs. But they did discuss that this GLI1 gene is a transcription factor responsible for controlling the activity of lots of other genes. So researchers were like, well, let's find another one that's maybe a little bit more precise to what we're looking for. So then they identified another gene that they named the Schistosoma mansoni non-ribosomal peptide synthetase, for short SMNRPS. And this one streams amino acids together in short peptides, and this one is essential for male worms to induce female sexual development. And this gene actually also develops and makes a pheromone, which is called the B-alanyltritamine pheromone, and this pheromone that this gene produces also stimulates female development. So without it, they wouldn't be able to reproduce, and then there would be no eggs. And I did find this picture of the paired worms. So right here is the B-lanoltryptamate, the pheromone that I was talking about. So SMNRPS makes this pheromone, and then this is what helps make those eggs. So these pheromones, these two things get together, B-alanine and tryptamine get together, they make this pheromone, and then this produces the eggs. So they're trying to find a way to separate these two things so that they won't get together, and then there'll be no eggs, is a future treatment that they're looking to make. And then here is the preclinical study that I found. They do discuss the mass use of the drug that we have already discussed. And again, they bring up the detail that, yes, it's safe and efficient, but it does not prevent reinfection. And with reinfection, you need to keep taking the treatment. But if you keep taking the treatment, there is a possibility that they can become resistant to the treatment. So, all right, so the main strategy that they are looking for in this study is reverse vaccinology. And this is just a strategy that goes into, they look at the whole process of the disease, and then they're going to use the mechanism of the disease to create a vaccine. So they used T cells derived from invasive stages of pear worms. So they went, if we look right here, these are the T cells right here that they're using in the middle of the whole of the development of the eggs. So they're going to use T cells from that to make vaccines, or an attempt to make a vaccine. They found target molecules that were consist of designing synthetic peptides from these proteins expressed during critical stages of the parasite's life cycle. So it's the T cells that they're going to be using from the most invasive stages of the pear worms. So they used 7-week-old female mice. The mice were kept in standard conditions in their experimentation facilities. The health and welfare of these mice were monitored during all experiments for pain, and all efforts were made to minimize animal suffering. They also used freshwater snails, who were used as intermediate hosts, and then these freshwater snails were infected with the Maricadia, which is the larvae before this is when the eggs hatch. So that's what they infected the freshwater snails with to make these cercaria, which would then infect the mice. And they used 3 T cell peptides that were synthesized, and they were made for the vaccination used on the mice. And the T cells were derived from the S. mansoni species of the snail. They came to the conclusion that the liver of the vaccinated mice significantly reduced affected liver surface than the unvaccinated mice. It looks like these T cell peptides were an important part of the protection, and they could be candidates for a multi-antigen vaccine. This is the main piece that they found here, that the liver surface was unaffected in vaccinated mice versus the unvaccinated mice. These results relate to our disease because this is another step closer to a vaccine. They have this pathway that has, seemed to have a positive outcome. And definitely, I feel like we will be hearing more about a vaccine using this method. Yeah.